This slideshow will cover the third part of our lecture on disease principles, and the major focus of this lecture will be on viral and bacterial infections. This slide talks about the 5 basic stages of diseases, specifically for communicable diseases. In the first stage, called incubation, this is the point of time where the disease or agent enters the body, so the pathogen enters the body, and the first symptoms occur. Sometimes that's a week, sometimes it's months, depending on the pathogen. Sometimes in incubation phase, people do become contagious and the disease can spread. In the prodromal phase, there are general symptoms that occur, and all of us have experienced this, and this is where there might be a day where you just don't feel well. You might have a slight fever, a headache, a scratchy throat, you might be nauseated, your nose might be running, and you know you don't feel well, but it's not clear that you're definitely getting sick. You may feel like, oh, I don't feel well, I'm going to take some ibuprofen or some cold prevention medications that are over the counter now, but it's not really clear that you're sick. However, in this stage, you are contagious and the disease is highly communicable. During the clinical phase, the characteristics and symptoms begin to appear, and this is where the peak occurs in the disease, and it's where you are most likely to spread the disease. When looking at these 3 stages, what's really important to understand is that often people are contagious or can spread the communicable disease with having no or little symptoms of the disease. And so that's why prevention becomes so critical when we look at communicable diseases. During the 4th phase, it's called decline, and this is where symptoms begin to fade and the person begins recovery. However, relapse can occur. I'm going to give you about 10 seconds to think about why might relapse occur. I will give you one example, and there are several, but I'm only giving you one during the slideshow. One example occurs with bacterial infections. Oftentimes with bacterial infections, a person is given antibiotics. Sometimes when people begin to feel better, they forget to take the medication, or they just decide, I feel better, I'm not going to finish the entire prescription. When that occurs, if recovery has not been completed, then you can relapse and the bacteria can begin to reproduce again and the person can get sick again. And that's one example of how relapse can occur. In the last phase during convalescence, the body heals and the host can become immune to the pathogen. Sometimes even though the host becomes immune, it can still be transmitted. And so that's an interesting thing to consider as we look at the variety of diseases. So these are the 6 types of organisms that we're going to study in the first half of the semester. And in this slide presentation, we're going to look at the bacterial and viral infections specifically. These are some infectious agents for bacterial examples. So bacteria is a single-celled organism and they're abundant in our environment. However, most of them are harmless. When they do cause illness or pathology, they're often treated with antibiotics. So there's 3 common groups of bacteria. The first one is rod-shaped or bacilli. There's tuberculosis there. There's round-shaped or cocci, which is pneumonia. Gonorrhea is also this type. And the last one is spirillum or spiral, and an example of this is syphilis. So we're going to look at 2 different bacterial infections and try to apply everything from the other 2 slideshows to a disease so you can see how everything connects. The first disease to look at is strep throat, and the agent is group A strep. The reservoir is the human throat. It exits through mucus. It's transmitted through airborne, indirect, or direct. And the reason why I have indirect or direct is some people list airborne as direct transmission and some people list it as indirect. It enters the host through the mucous respiratory system, and the host is human, and they're younger than they are older. So that goes through the chain of infection. Here are the steps of prevention with strep throat. In primary prevention, we're trying to prevent the disease from occurring. So this is where you might teach people to sneeze into their arm, to wash their hands often, to not put things like pens or pencils in their mouth. And so those are all examples of primary prevention with strep throat. In secondary prevention, these would be interventions that lead to the discovery and control of the disease. So for example, in this case, the person might go to the doctors or the clinic and get a rapid strep test. If it comes back positive, then they're probably put on antibiotics, and that is an example of a secondary prevention. Tertiary prevention occurs when we're trying to prevent the impact of the disease from having a long-term effect. So let's say that someone has strep throat and it's not treated correctly. It can then actually spread to other parts of the body and might cause something like rheumatic fever, which actually causes heart problems. So in order to prevent that disease from having long-term effects on the heart, the rheumatic fever would be treated, and if treated correctly, they could at least stop the progression of damage to the heart. So those are some examples of how to apply the first two slideshows to strep throat. The second bacterial infection we're going to look at is syphilis. The agent is Treponema pallidum. The reservoir is human. It exits through mucus or body fluids. Usually it exits through sexual fluids. Its transmission is through body secretions, typically sexual secretions. It enters the body through mucus, body fluids, or through tissue. So if someone has syphilis and they have oral sex, someone performs oral sex on them, the bacterial infection would spread through the mouth, and the primary symptoms might occur in the throat. So it's sexual fluids usually, but sometimes those fluids enter other parts of the body other than sex organs. And the host is a human. One thing to consider with spread of any sexually transmitted infection is if your person has an STI already, it alters the makeup of the sex organs, which actually makes it more likely for them to get a second STI. These are some issues with prevention. So prevention means that we're preventing the disease from occurring, and with syphilis, that would mean using a condom every time someone has sex. And if it's oral sex, using dental dams or still using a male condom, or if it's vaginal sex, using a female condom or a male condom, and in anal or intercourse, using a condom. The secondary intervention that leads to discovery and control of the disease would be getting a blood test to see if the person has syphilis. And if the person does have syphilis, they would be given antibiotics in order to resolve the issue and have syphilis be cured. Sometimes, however, people do not treat syphilis. They don't realize they have symptoms and therefore don't get treatment. And syphilis is one of the sexually transmitted infections that can spread to other parts of the body. So in tertiary prevention, in the case of syphilis, what happens is people actually may have damage to their nervous system or their heart, and then those conditions have to be treated so as further damage is not done. So now we're going to move on to viruses, and viruses are one of the smallest of the disease organisms. In reality, they could not even be seen until the electron microscope was invented in the 1930s. An important issue about viruses is they are comprised of DNA or RNA, which is a protein. And viruses cannot live outside a host. They may be dormant, but they cannot live and reproduce outside of a host. They're able to penetrate cells, and they use the cell's nucleic acid to produce more amino acids, and they often reproduce and take over the cell of the host. And that's why a host can be infected but not have the disease. So it depends on how the virus acts within the human cell. Later, an outside factor, for example, lifestyle habits or some other physical illness or infection, can actually activate the virus that's been dormant in the human. When the virus bursts out of the cell, they destroy the cell in which they were living, or they degenerate that cell, and they move on and infect other cells in the human. And viral infections are more prevalent than viral diseases, and we'll look at a couple viral infections now. So the first viral infection we're going to look at is influenza. And if you look at the chain of infection, the agent or pathogen is the influenza virus. The reservoir is the human respiratory system. It exits the human through mucus. It's transmitted airborne, and some people consider airborne direct and some people consider it indirect, and that's why both terms are there. In this class, it's basically considered indirect airborne. It enters the host through the mucus respiratory system, and the host is a human, and younger, older, and immune-compromised people are more likely to be infected by this virus. If we look at the prevention levels, primary prevention prevents the disease from occurring, and this might include vaccination or the flu shot. Secondary interventions lead to discovery and control of disease so it doesn't get more serious. And so when someone has flu-like symptoms, they may go to a doctor or an emergency room, and it is identified, and they may be given medications to help with the symptoms. Sometimes there's antiviral medication they can take if they go early in the disease process, but those are all designed to help the disease from getting into other illnesses or progressing and making it more serious. In tertiary prevention in this case, we're looking at the prevention of flu causing other long-term effects on people or having chronic conditions. So sometimes the flu turns into pneumonia or chronic bronchitis, and then we have to treat those conditions so that there's not permanent long-term effects. And so those are examples of how the levels of prevention work with the flu. And the next one we're going to look at is chickenpox, and the varicella is the agent. The varicella virus is the agent. There's a variety of varicella viruses. Chickenpox, the reservoir is the human respiratory system. It exits through mucus. It's airborne. It enters the host through the respiratory system, and the host is the human, and the host has to be unimmunized or immune compromised. And so that covers the chain of infection for chickenpox. Primary issue here is the prevention of the disease from occurring, and there's varicella vaccines. The intervention that leads to discovery and control of the disease, if someone has symptoms of chickenpox, then they might go to the doctor. Typically, chickenpox has to take its course, but often if they go early enough in the disease process, they can go on antiviral medication to make the disease less severe and last less time. And tertiary is to try to prevent the impact of disease from having a long-term effect on people. And this is going to be an example of where an immunity can occur, but the virus lays dormant in the person for years. And that is in the case of diseases that might occur later in a person's life. And so if someone has chickenpox, they can then come down with other diseases later, even though the chickenpox virus was dormant in them for a long time. So here is a comparison of bacteria and viruses so you can see the two and why they're different. So if we look at them, and I'm going to go side by side instead of going down. So in bacteria, bacteria can live outside the host longer than viruses. They can't live outside the host indefinitely, but they do live outside the host longer than viruses do. Viruses have a very difficult time living outside the host because they need the cellular structure in order to survive. Bacteria are treated and cured with antibiotics, and viruses are not. In a viral infection, typically the disease is not treated, but the symptoms are treated. And we looked at a couple samples in the prior slides that show that. Bacterial infections typically have low-grade fevers. Viruses typically have high-grade fevers. In adult humans, that's over 100 degrees. In children, a high-grade fever is higher because they're able to deal with the fever better. In bacterial infections, there are rarely immunizations for them. In viral infections, immunizations are a primary way of preventing the spread of the disease. In bacterial infections, the symptoms are often localized to a specific part of a body, for example, strep throats in the throat. In viruses, the symptoms are more generalized because the virus spreads throughout the body, and then you have symptoms that occur throughout the body systems. So those are the comparisons of bacteria and viruses, so you can see very clearly how the two of them are different from each other. So that concludes part 3 of our look at principles of disease. You have some online assignments to do, and as you're doing your online assignments, I'm encouraging you to use the actual terminology. For example, airborne, indirect, direct. I want you to use the actual terms of epidemiology so that you get used to talking about diseases in those terms. So thanks for watching the slideshow, and it's time to move on to your online assignments.